All right, so I'm going to talk to you about understanding the role of RNA interference in endogenous gene regulation. So there's a lot going on there. But really the basic question we're trying to answer in the lab is how do organisms determine when genes are turned on and when they're turned off? When you ask it in that broader sense, it's a little bit easier to understand. And so we're going to use our model organism, C. elegans, to help to understand this question. And I will show you some of the work we've been doing in our lab to answer this. So some of you are probably familiar with this. This is a central dogma of molecular biology. And it really represents how information flows within a cell. Genes in DNA are transcribed into messenger RNA. These messenger RNAs are then translated into proteins. Now, proteins are kind of the versatile functional molecules of biology. They're the enzymes that catalyze reactions within our bodies. They're the structural proteins that make up our fingernails and our hair. And but one of the really interesting things about biology is that every single cell within our body has the exact same DNA, but it doesn't necessarily have the same protein. So how do we determine what genes are turned on when in our bodies? It's really a central question to understanding biology. And really organisms accomplish this regulation on a number of levels. First, we have transcriptional regulation. You're preventing DNA from being transcribed into messenger RNA. You can also have translational regulation, or post-transcriptional regulation as it's sometimes referred to. This is when messenger RNA is prevented from being translated into protein. The pathway we work with in our lab, known as RNA interference, actually works on both levels. So it's a very powerful pathway, it's a very specific pathway, which makes it very exciting. Our pathway really begins with this molecule, double-stranded RNA. You're all probably familiar with DNA, double helix, complementary base pairing, A's with T's, C's with G's. A lot of the same things are going on with double-stranded RNA. You still have complementary base pairing. You still have a helix, albeit a slightly shorter and wider helix. Now, a lot of you are probably familiar with RNA. You're thinking of it in terms of a linear molecule. You don't have two strands, you have a single strand. And that's definitely true. For messenger RNA, that's true. But really, the more we learn about RNA, we're learning that most RNA has secondary structure. It's a much more dynamic molecule than we first thought. So what does this double-stranded RNA do? It induces this pathway known as RNA interference. It's cleaved by this enzyme known as dicer into smaller double-stranded fragments known as small interfering RNAs. These are about 21 to 23 nucleotides long. And they associate with a number of different protein complexes. One of these protein complexes is known as RISC, or the RNA-induced silencing complex. siRNAs in RISC can associate with messenger RNAs. And once that association occurs, RISC can actually catalyze the cleavage of a messenger RNA, which you see in the bottom here. Once this messenger RNA is cleaved, it can't be translated into protein. Effectively, you silence this gene. Really, one of the critical things about this pathway is this interaction between this small interfering RNA and this messenger RNA is dependent on complementary base pairing. In other words, these siRNAs target specific genes within our bodies. That makes it a really, really powerful pathway because as scientists or even as organisms, we can target individual genes. Now, the slide I just showed you is really a skeleton picture of RNAi. There's a lot more going on. There are many more proteins involved. And really, we haven't even found all the proteins yet. But that's one of the questions we're trying to answer in our lab. What genes, what proteins are involved in this process? So we designed this screen to really ask that question. We work with C. elegans. Some of you may be wondering, why C. elegans? It turns out that C. elegans share a lot of the essential genetic components that we have. A lot of the genes we work with have direct human homologs. So a lot of the questions we answer in C. elegans have direct, direct bearing on human biology or the workings of higher organisms. And so we screened the entire first chromosome of C. elegans using this transgenic array depicted in the left. We used the gene for green fluorescent protein, some of you may be familiar with. It's isolated from jellyfish, and when you shine UV light on GFP, it fluoresces a bright green, which I guess its name implies. These worms also contain an inverted repeat of GFP attached to a heat shock promoter. And so what this does is under normal conditions, these worms fluoresce bright green. It's a very visible phenotype. It's really nice to see. When heat shocked, worms that support RNA interference lose fluorescence. You get transcription of this inverted repeat of GFP. Because it's an inverted repeat, there's complementary base pairing. It forms double-stranded RNA. This double-stranded RNA induces an RNAi response that quenches green fluorescent protein. In worms that don't support RNA interference, it remains fluorescent. So we turn something from a very molecular level into something we can see under the microscope, which is very nice. It's a really high throughput screen. As you can see, we tested 1,800 different genes. So what are the results of this? What are the results of this screen? Here's kind of a better picture of what's going on there. These are actual worms. You can see the green fluorescent isolated in certain cells here. On heat shock, you lose almost all fluorescence. However, if you have a defect in an RNAi gene, dicer 1 here you saw on that initial overview of the RNA interference pathway is required for double-stranded RNA processing. If you have a mutation, dicer 1, you heat shock these animals, these worms stay fluorescent. So you can see the difference between these two animals, and it really is that clear when you're looking under the microscope. Is RNA interference working in these animals, or is it not? So we got some interesting results from this screen. We pulled out this novel gene SOP3, which I'll talk about later. We identified MUT16 and SMUG1, which were independently identified in other labs as playing a role in transcriptional gene silencing through RNA interference. We pulled out a couple other genes, a zinc finger protein, they're usually involved in DNA binding, an ATPase, kinesin-like protein, and several additional genes. For the purposes of this talk, we'll focus mainly on SOP3. So one of our first goals was to confirm the results of that initial genetic screen. So we obtained a mutation in SOP3, introduced it into animals containing our transgenic array, and heat shocked them. We basically repeated our initial experiment, but with a mutation, not necessarily just a knockdown of that gene. We got the same data, which is always encouraging. SOP3 animals, when you heat shock them, remain fluorescent, indicating that RNAi is not working in SOP3 mutants. Why are we interested in SOP3? Well, SOP3 kind of piqued our interest because before we picked it out, it was known to interact with a class of genes known as homeotic genes, or HOX genes. Homeotic genes specify a location within an organism and lead to the development of structures appropriate to that location. So here is actually a really dramatic and kind of nice example of homeotic genes in action. Here we have a normal wild-type fruit fly, and on the right here we have a fruit fly with a mutation in the homeotic gene. And you can see right here he's actually got legs growing out of his face where antennae should be. And so that's how powerful these genes are. And here we have RNAi controlling this pathway. It's kind of an exciting phenomenon. And but what exactly does SOP3 do within C. elegans? So SOP3 was initially identified as a suppressor of PAL1 SOP. And PAL1 is the actual homeotic gene in this cascade, and it leads to the development of these organs known as V6 rays. V6 rays are these kind of finger-like projections coming out of the tail of this animal. When you have a mutation in PAL1, you lose these rays. You can see the difference between the top figure here, a wild-type animal, and a PAL1 mutant. You lose those rays. And SOP3 is suppressing a pathway that can turn on PAL1 even in the absence of a PAL1, even in the presence of a PAL1 mutation. When you get a mutation in SOP3, however, this Wnt pathway can independently activate PAL1 and lead to the restoration of rays. So this is a little bit funny. You have to step back and say, so we have a mutation in PAL1, we lose rays. We have a second mutation, and we gain rays. What exactly is going on there? And kind of more generally, what does this have to do with RNA interference? We know that SOP3 is involved in RNA interference. We know that it's involved in regulating PAL1. How are these related? Is SOP3's regulation of PAL1 a consequence of that RNAi activity? And so to answer that question, we looked at other RNAi defective genes. We'll look at dicer, which we saw earlier. We'll look at a dicer-related helicase and see if we introduce mutations in those genes into a PAL1 background, whether we get similar suppression as we do with PAL. And the answer is sometimes yes, sometimes no. So you can see in a PAL1 mutant, only about 5% of animals have these V6 rays. It's a really low percentage. When you introduce a mutation in SOP3, you suppress that mutation, and 84% look wild type. 84% have basically normal rays. If you knock down dicer1 via microinjection, you get about 50% suppression of this PAL1 phenotype. And if you knock down DRH3, which again is a dicer-related helicase, it has a very similar phenotype to dicer. You get, again, about a 50% suppression. So this is, to us at least, it's very interesting, because you have RNAi that seems directly implicated in the regulation of these big homeotic genes. RNAi is leading to body pattern development in these animals. And so we have a lot of questions to answer. You saw this transcriptional cascade. You know, PAL1 was turning on MAB5, was turning on EGL5. And we know that RNA interference is acting somewhere in that pathway, but our data doesn't necessarily nail down where that regulation is occurring. So that's a big question to be answered. You saw the Wnt pathway involved there. The Wnt pathway is huge. It's one of the first really direct links between genetics and cancer that we have. Mutations in the Wnt pathway had a dramatic effect in colon cancer in humans for a long time. So really we need to understand this pathway better. And it's been a lot of work. And you know, presented this way, this data looks very nice. We've also tested really many other RNAi defective genes, and some suppress and some do not. What do the genes that suppress this phenotype have in common? What do the genes that do not suppress have in common? So there are a lot of questions to be answered here. Really a lot of work to be done. But you're probably wondering, you know, how does this all relate to the greater field of biology? Why should I care? And so the answer is RNAi is really a promising technique. People have thrown it out there as a possible cancer therapy. Some promising data a couple of years ago linked RNAi suppression to reducing transmission of HIV in cell lines. They think it may be able to treat dominant genetic disorders. In terms of research applications, it's a huge tool. The ability to knock down a single gene is like a researcher's dream. But to really effectively utilize this tool, we have to understand what's going on. And so that's what labs like ours do. We are kind of understanding the underpinnings of this pathway so we can effectively utilize it. And really, on a broader, more basic science level, we've seen that RNAi is involved in a lot of gene regulation. You know, we just presented one example here, but estimates kind of vary. Probably about 30% of human genes are under some kind of RNAi control. So really, just understanding how our genes are regulated is very connected to RNAi. And I will just finish off by acknowledging my lab members. I'd like to thank Dr. Knight, and that's me again, Eric Gabriel, who's a junior in the lab now, Jeff Hespy, who graduated last year, Leah Malazzo, who is a fellow senior, and our past lab members, Lori Spicer, who's still here but works in another lab now, Kristen Robinson, who was at the NIH for a while and is moving on to pharmacy school, Jeffrey Kummel, who is going to be a dentist, he's at MCV now, and Xavier Belcher, who's also at MCV, going to be a doctor. Unfortunately, we don't have a picture of Annie, who is the latest member of our lab, but she is also with us. And thank you for coming, and I'll be happy to take any questions.